Translation. Building the protein translation is the second stage of gene expression, where the mRNA sequence is used to build a specific protein. This process takes place in the cytoplasm, where ribosomes, the cell's protein factories, bind to the mRNA. The ribosome reads the mRNA in groups of 3 nucleotides called codons, each of which corresponds to a specific amino acid or a stop signal. Transfer RNA molecules play a critical role in translation. Each tRNA has an anticodon that is complementary to a codon on the mRNA and carries a specific amino acid. As the ribosome moves along the mRNA strand, tRNA molecules bring the appropriate amino acids to the ribosome. The ribosome then links these amino acids together using peptide bonds, forming a growing polypeptide chain. This process continues until the ribosome reaches a stop code, such as UAA, UAGA, which signals the end of the protein synthesis. Once the polypeptide chain is complete, it is released from the ribosome and begins to fold into its 3-dimensional structure, becoming a functional protein. This folding is crucial, as the shape of a protein determines its function within the cell. Some proteins may also undergo further modifications or be transported to specific parts of the cell to perform their tasks.